People appear to be having more fun at the posters than they should be. All right, I will get started. This is part 3 of the 4-part course that we are all part of. And I will begin today with the scope of what we will talk about today. We'll begin with a story so far, and then I'll launch into this collection of short stories that I want to share with you. Each of them kind of can stand on its own. I'll talk about rectification. I'll talk about active Brownian particles inside walls and bags. And I'll make a brief detour to talk about interacting ABPs, just to make some comments on MIPS, because we've talked about MIPS quite a bit in the discussion. I wanted to add my 2 cents worth to that. And if we have time, we'll do some philosophizing at the very end of the lecture. Okay, let's start with the story so far. We have considered 2 irreversible processes, diffusion and damping. I argued that both of these are irreversible, namely that if I showed you a movie, you can tell whether the movie is going forward or backward, and both of them arise because of my absence of knowledge about what the water molecules are doing. And we built some math to try to describe this phenomena. We talked about Langevin equations, Fokker-Planck equations, and time correlation functions. And what should have been fun, and you can tell me whether it was fun or not, we did 2 problems. One was diffusion of a passive ellipsoid so that we can have practice dealing with coupled Langevin equations, and dynamics of an active Brownian particle so that once in your life you can do the full calculation of deriving an ABP's MST. And then we started talking about an active ideal gas, and we considered one particular phenomenon, which is the pressure of an active ideal gas. And then I asked you to look up some literature associated with what's been happening with pressure over the past 10 years. What today is a collection of stories about active ideal gases. I will not be showing too much of the math. I will be trying to tell you stories and sketch cartoons and so on. And then I will point you to the references where you can go and look up the math. And I will basically force you to look up the references a little bit in the part of the exercise segment of the class. And I have a little low energy today because I'm grumpy. And you guys seem to have low energy today because you had too much fun yesterday or something. So we both have to boost each other's energy up to get through the next hour and a half. Okay, I'm launching into my first story. My first story is rectification. What I want to do is I want to take non-interacting ABPs, the same thing that I was looking at when I was thinking about the pressure, and I want to put them in a box, and I want the box to be of this form, where the box has 2 sides, and the 2 sides are separated by these wedge-shaped openings. Now, let us begin by asking, what would I expect to happen if I put passive colloids in equal concentration on either sides of this box? I would claim that I could put the passive colloids in this box, and I could go away, have a cup of coffee, get married, make a baby, come back. I would still have half equal concentration of active colloids on either side. Because if spontaneously there was a gradient that developed in this problem, then the second law would be wrong. And the way that it works for passive colloids is in equilibrium. The system is extensive, the shape of the wall does not matter, the size of the opening matters, and whatever fluctuations result in particles going from this side to that side will be compensated by particles going from this side to that side, so the net current will be 0 on average. So an equilibrium system inside such a box would always stay where you started it out in terms of concentration. But now, if you decided to put self-propelled particles inside this box, we ask ourselves what happens, and what happens is demonstrated in this experiment. This is an experiment done in Bob Austin's group many years ago, where they put E. coli in equal concentrations on either side of this box that is there in my cartoon, and you wait about 20 minutes, you start building up a concentration gradient, with most of the E. coli ending up in 1/2 of the box. This is the phenomenon that has been called rectification in the active matter literature. And this paper is the reference to the original experiment, the first experiment in this category of experiments. There are a lot of them now. Okay. So what I want to do for the next 10 minutes or so is to talk about this rectification phenomenon. Let's begin with some context in non-equilibrium statistical mechanics. And to place that context, let's consider the thermal colloid problem that I started with. What I mentioned is that for the thermal colloid, this shape does not matter. The size of this opening is the only thing that matters. Supposing the size of this opening is A, flux of particles that will go from this side to that side will again be proportional to the size of this opening A. So these two fluxes will cancel, and I will always have equal concentration of particles on either side. This, in a cartoon way, for us non-equilibrium stat-mech people, can be represented by considering a particle that is subjected to white noise, so a Brownian particle, that is diffusing in a ratchet potential. A ratchet potential is basically a guy like in the cartoon over there, where you have this triangular potential shape, and you have a Brownian particle that's trying to move from one well to the other. It can either jump to the right well, or it can jump to the left well through Brownian motion. Okay? And since the barrier height is the same on both sides, the particle is equally likely to go this side or that side. And therefore, if you decided to make an experiment in which you watch the particle jump about in this triangular potential, and I showed you the experiment movie in reverse, you can't tell where I started the movie and where the movie ended, because it's equally likely to go this way or that way. Does this make sense? This kind of modeling of the rectification phenomena falls under the category of Brownian ratchets, and the steady state current of this model will correspond to the flux from one chamber to the other. So this model can be a theoretical idealization of this problem that we were talking about, namely I put E. coli in a box and most of them end up on one side. Okay? This would be the mathematical framework one would use to try to understand this problem. Now let's place a little bit more context in terms of what it is that I'm saying. Rectification is a term that was coined in the context of E&M, right? Has everybody built rectifying circuits at some point in their life where you put an LC circuit such that you convert an AC signal into a DC signal? That's what rectification is, that we are talking about here. What we are saying is my drive is AC, namely it's equally likely to be in this direction or that, but my response is DC. It always goes in one direction. That's why this phenomenon is called rectification. My drive for my self-propelled particle is random. It can go in any direction it wants. But my response to the presence of this wall is that I end up going in one direction. So that's the origin of the name. And there is a rich history in non-equilibrium statistical mechanics for Brownian ratchets. The first ratchet that was proposed was the Smoluchowski ratchet. It's now called the Smoluchowski-Feynman ratchet because people briefly forgot about it for 50 years and then relearned it when Feynman included it in his lectures. So they are called, it's called the Feynman-Smoluchowski ratchet. But I'm really not going to spend time telling you about pawls and gears and things like that. The standard theoretical construct that one uses to try to understand these kinds of phenomena is to use triangular potential and put a particle in that triangular potential and compute the flux of which direction the particle is likely to move. This is the standard calculation that one does. And I encourage you to play with ratchets. What I mean by that, it's a really toy problem. If you like solving equations, ratchets are the prettiest problem that you can solve. Most of the math is tractable. You can do a lot of work analytically, and the numerics is dirt cheap. You can do it on MATLAB or something in 15 minutes. So it's really fun to explore non-equilibrium stat-mech in the context of these models. And I'm going to claim, but not prove, that the system has to break spatial symmetry and time reversal symmetry in order to get a current. I have to somehow set up my ratchet potential and my particle in such a way that the system breaks both spatial symmetry and time reversal symmetry. Now, let's pause for a moment and just understand what those words mean. I'm not going to prove them, but I want to understand what those words mean. So let me consider my symmetric triangular potential. In this potential, the left direction and the right direction are the same by the construction of my potential, right? There is no speciality to left or right in the construction of this potential. So this potential has spatial symmetry. And if I put a Brownian particle subject to white noise, that is delta function correlated noise, then I have time reversal symmetry as well. Markov processes are time reversal invariant. So there is time reversal symmetry as well. So the problem that I started with by saying, oh, this is a ratchet problem, that has both spatial symmetry and time reversal symmetry. And therefore, this model will never result in a current. Now, I can break spatial symmetry easily enough by changing the form of the potential. I can change this triangular potential into a sawtooth. Now there is an easy direction and a hard direction, right? The particle feels less force if it's going this way and feels a much bigger force going that way because the potential is steeper. Right? So I broke spatial symmetry by changing the shape of my potential. And this is how people always break spatial symmetry when they're talking about ratchet models for rectification. But breaking time reversal symmetry can be tricky. So there are a few different things that people do when they want to break time reversal symmetry. The first one, which I call stupid and easy, is tilt the potential. If supposing I put an overall tilt on this potential so that this potential was actually going on this line, and I showed you a movie of a ball climbing up this hill, you know I'm showing you the movie in reverse, right? So this is the stupid way of breaking time reversal invariance. You just tilt the potential. Because now if I make a movie of what is happening here, you can all tell that, oh, the ball, you're showing me the movie in reverse. What is the ball going up the hill? Right? So tilting the potential is one option, which I call stupid and easy because you know, this is really stupid. And easy because the math is super easy because you're just shifting the potential by a constant straight line. So the calculation is very straightforward and you can do everything. The next best thing that people have considered in terms of breaking time reversal symmetry, this is somewhat clever, is to have a fluctuating potential. In the cartoons I'm drawing here, the potential is independent of time. This is just fixed. It's this guy, right? What you do is you change this potential so that it becomes a V of x comma t in such a way that the potential turns on, turns off, turns on, turns off. You make the potential turn on and off. So you turn on a fluctuating potential. In this case, you can have a fluctuating potential, and the fluctuating potential does not, you cannot, if you just had the potential, you can't tell whether there is no time reversal symmetry breaking. If you had a Brownian particle sitting by itself, there is no time reversal symmetry breaking. But you put the two together, time reversal symmetry gets broken. So that's one way to get rectification. But my favorite way is use colored noise. And you will see in a minute why it's my favorite way. And I want the bottom paper, the PRL, is the one that introduces colored noise for the first time in ratchet potentials. And these two papers on this side of the slide are the papers I recommend you look at to get a sense of what the role of Brownian ratchets is in all of the stuff that now belongs under the umbrella of active matter. But before there was such a thing called active matter, people were playing with ratchets. And there's a huge body of literature, and these two papers offer a starting point. And the reason that I'm recommending these literature to the young people is it's a treasure trove of tools. There's a lot of nice math that people have already worked out that can be easily borrowed, and there are infinitely many active matter problems to which they can be applied and have not been yet. Okay? So you guys can do all that. I'm too old and lazy, so I'm not going to do that. Okay. Now, we'll pause here for a moment. If you are able to scan this QR code, it will take you to a Google Drive folder. Yes, sorry.
I have a question.
Yes.
Why the two effects together break the time reversible symmetry? I didn't get why.
Ask me one more time.
Why the two effects together break time reversal symmetry?
Oh, okay. I didn't tell you. The reason you didn't get it is because I didn't tell you. It's hidden in the math, and I don't want to talk about the math, so you're going to have to read the paper. There is no simple story explanation. It's just the way the math, the fluctuations here and the fluctuations there talk to each other in a way that you can break time reversal symmetry. It's associated with having two sources of fluctuation, one in the potential and one in the particle. Okay? And unfortunately, there's no cartoonish hand wave argument that can be made for this, or at least I don't know how to, so it's in the math. Okay, can I ask if you can scan this QR code? If you do that, you should go to a Google Drive folder. Can you try and tell me if it works? Yes, okay. So now let's switch to the Drive folder. So if you go to this Drive folder, there is a document called rectification exercise. Do you guys see that? Yeah, okay, double click on that and open that. And if you scroll up, let's see, where is my slides? Yeah, I would like, within the, okay, so if you go back to the folder, where's the folder? There is a file here for the Brownian ratchets in physics and biology paper. That's one of the papers I recommended. There's a PDF file in that folder. Okay, open that file and do the following. Find the figure I should look at to learn about a theorist's ratchet, a man-made ratchet, and a natural ratchet, a biological ratchet. I'm just opening the paper so I can scroll to the figure after you guys tell me. Take a look at all the figures. Think about it for a moment. Is everybody able to access the file? Good. Oh, QR code, yes. Now? Look in that paper, the PDF called Brownian ratchets that's there inside the drive. That paper is not always accessible online, so I put the PDF inside the, it's a very famous paper, but for some reason, the access online has always been questionable. The Brownian ratchets in physics and biology by Barr. You got it? Good. Okay. Are you guys crawling and looking at all the figures? Yes. I just want you to find one figure that corresponds to a theorist's ratchet, one figure that corresponds to a man-made ratchet, that is an experimentally realizable ratchet. You can't make a Smoluchowski-Feynman ratchet in reality. I assume everybody knows, right? You can't get to a low enough mass scale that you can get that thing to fluctuate. It's really a theorist's ratchet. It was made in Smoluchowski's imagination is basically it. Okay. Figure one, clearly theorist. Anything else? 7. Okay? Are there others that are theorist ratchets? I'm going to look at 7 to remind myself what it was. Ah, yes, 7. How about a man-made ratchet? I'm hearing 2, 6, 5. What else did I hear? There was some other number. 3. Okay. Let's look at those figures. So let's start with figure 2. Oh, figure 2 is a theorist ratchet. I disagree. What about figure 6? Six is man-made, yes. That's a good scheme. What is 5? Oh, 5 is also the electric field thing. That's fine. What was 3? Oh, yeah, okay. It is a man-made ratchet. Fine. Biological? Yes. 1 belongs in the man-made? Yeah, theorist is a man. So yes, it is man-made. Only you can't make it. Yes, I agree. Because it was Smolichowski's imagination. People have been trying to construct this Paul and gear ratchets for, I don't know, however long before Smolichowski said it would happen. Because it's very hard to build. Yes. 9 is also theorist? Probably. What is 9? Sure. There is no data on it that I can see, so it's also theorist. If there is data, then I would disagree, but there is no data on that plot. Say again? 8 is man-made, okay. Let's see what 8 is. Ah, yes. Okay. Now, biological. Is there any motor picture? 4. What else? That's it? 2. What was 2? I said it's not man-made. It was theorist? Okay. If I said it was theorist, then it was theorist. I'm a theorist. I'm allowed to name other theorists. All right. I just wanted you to get a flavor for this paper by forcing you to scroll through it, because this paper is very readable. Gives you an access to how to think about Brownian ratchets without too much of the math. And if you're biologically inclined, there is ATP with square brackets around it and things like that happening inside the equations. So that's fun. Okay. Let's switch back to the presentation. All right. Moving on. Second part of the exercise. Stay with the same folder. Inside it, there should be Charlie Doring's PRL. And there is a Google Doc that's called rectification exercise. Yeah? Put your quick and dirty 1-line to 3-line summary of what's happening in that paper in that Google Doc. I'll open the Google Doc. We'll see what you guys write. And you will be anonymous, so feel free to write whatever you want to write. In this Google Doc, under the thing where it says PRL by Doring et al., put down your 1-line summary after you scroll through the paper a little bit. Look at the Doring paper, scroll through it. It's one of my favorite papers. Charlie Doring was awesome. It's interesting to read all of his papers, actually. He hasn't written that many, so it's easy to read all of his papers. Unlike people like us today who write 1 paper a week, and it's impossible to keep track. It is a theory paper, but try to scroll through it and see if you can discuss with your neighbors, friends, enemies, whoever, and come up with some one-line statement of trying to make a sense of what is the paper doing. This is good practice. This is an essential skill for you to survive in research. Yes, sir. No, okay. The more you practice your skill of being able to quickly, in 10 minutes, decide what a paper is about, the more you're likely to read papers, your knowledge goes up, and the number of different views you can take to your particular problem goes up. So it makes you much more fun inside your head, I mean. I don't know what it makes you outside. Yes, sir. Oh, it's not allowing edit? Okay, sorry. Let's see, how do I do that? I apologize. So I've had this headache since yesterday afternoon, and so I am functioning at like 20% brain capacity. Share. Anyone with link, editor. Done. How about now? Does it work or not yet? Yeah, good, perfect, thank you. Sorry about that. Being stupid and in front of other people is very hard. Being stupid alone is okay. ChatGPT is not bad. Do you guys use ChatGPT to help with your writing? It's very useful, especially when you're writing something technical. The way I recommend that you use ChatGPT is first formulate your thought into bullet point phrases. Then use that bullet point phrase inside ChatGPT to make it into a pretty paragraph. Then read the paragraph and edit it. It's very useful, and it short circuits the writer's block that one gets when one is trying to make complete sentences. I do this exercise with my students where they're supposed to use ChatGPT to do their homework, but then they have to follow the rules and they have to put screenshots of what they did with ChatGPT. Other neural networks, other large language models that are not ChatGPT, I mean your brains. Let's see some original things here. Make room for yourself by clicking down if you like. I like the correlated noise statement. That's the one that I was looking for. That's why it's my favorite model. Let's wait one more minute to see if somebody else wants to add another statement, and then we'll move on. This is all my way of bribing you into reading Charlie Doring's paper. Okay. I'm going to switch out of this and back to the slides. Don't close the document or the drive yet. We are going to come back and do a couple more of these. And for the moment, switch back to me so that I can tell you some more stories. Okay. Now, why did we do this? Why did we look at Brownian ratchets and Charlie Doring's paper in particular? We looked at it because Charlie Doring's paper gives you all the math you need to. He never heard of active matter. Okay. Maybe he heard about it towards the end of his life, but he never heard of active matter when he wrote that paper. Okay. But he lays out all the math you need to solve the problem of rectification for active Brownian particles. Why is that? Because let's look at an active Brownian particle. This is my equation of motion. And you guys know that that u hat is cos theta x hat sine theta y hat, and that theta is subject to white noise. Yes? Everybody knows that at this point. And if I just call this v0 u hat, the whole guy, as some variable z, you should be able to prove for yourself. And this is part of the exercise that was handed out in the previous class, that the noise correlation, if you treat that whole thing as noise, is precisely exponential. It's a correlated noise with one frequency that's given by 1 over dr. Given by dr, I mean. You can verify this readily based on all of the Langevin stuff that you already know how to do or will know how to do when you finish solving the problems. You can show that this is what an ABP is. And this is precisely the calculation that is there in the Doring paper. So the noise is correlated, and therefore I can use the math that is there in the Doring paper and solve this problem of E. coli accumulating on one side. And that's the math. Let's take a physical picture. Why do self-propelled particles exhibit rectification in general? Let's look at the openings that we constructed between the two halves of my box. And let's look at what happens when a passive colloid collides with the wall. When a passive colloid collides with the wall does trigonometry, figures out what is the normal component of the force exerted by the passive particle on the wall, exerts an equal and opposite force, right? Newton's third law. If the passive particle is hitting the wall, the equal and opposite of the normal component will be the force exerted by the wall on the particle. So if I take that force, then the net force on my particle after the collision will be in that direction. Think about this for a moment. All I'm saying is the wall exerted an equal and opposite normal force, and I add this force and that force, and I get a velocity in that direction. So any passive particle colliding with the wall will end up with a velocity that turns it back from the direction in which it came. Okay? But if you put a self-propelled particle there, again the wall will do trigonometry and exert an equal and opposite force, and there will be a net force in that direction. But the particle will still keep swimming into the wall. Right? The self-propulsion velocity is self-replenishing. The self-propelled particle is stupid. It doesn't know about the wall. It only knows that it's swimming. So it runs into the wall and keeps running into the wall. So the net velocity is parallel to the wall. And therefore, what happens? When a particle comes from the left, it sees an effectively bigger hole than the particle on the right. Do you guys see that? The whole wedge becomes a hole because anybody who ends up in the wedge gets convected to the other side. So the hole size is different on two sides. Obviously, more people will end up on the other side. So this is the physical interpretation of this rectification mechanism. Okay? And when is this effect significant enough that I can actually measure a macroscopic concentration difference between this side of my box and that side of my box? That will happen when the time taken for my particle to transit the barrier, let's call this distance as L. This is the time, this is the size of my wall here, okay? And I ask, what is the time taken for me to move this length? That should be 1 over L over the speed of the particle, right? Yes? And this time, the time it takes for me to transit the barrier should be short compared to the rotational diffusion time. That is to say, I should keep pushing into the wall till I successfully navigate that distance. If I turn, then I'm screwed. Right? So I need the rotational diffusion should not kick in while it takes while I take the time to transit the wall. When this condition is satisfied, that is the circumstance under which you can get macroscopic density gradients building up. Okay? I can't tell if I'm grumpy or you're grumpy. Yes? Pardon me? No, it's quite continuous and it depends on your clock and ruler. You decide what size your system is and how much gradient you will consider as actual gradient. That depends on your ruler, right? How much concentration difference you can resolve. It's not a transition. It's a continuous thing that happens. And the exercise here is to find these two papers and put your one-line summary of these two papers into the document. I have my one-line summaries on the other side of this slide. I will put them there in a moment, but I'd like to see what you come up with. The active ratchets paper and this light-induced self-assembly paper. Find them. This is how much information I'm willing to give you about it, which is actually a lot of information in this age. These are two very nice papers that give you a handle on how to think about rectification in active systems. No, sorry, I was like, no, I'll let you guys do it. Otherwise, you'll have to write your one-line summary by looking at just the title and abstract on Google Scholar. That's okay. I'll switch to my Google Doc for a minute. There are a lot of rectification papers in active matter at this point. I counted about 25 the last time I checked, which was, I don't know, a year or so ago. So there are a lot of them. This is a very cool phenomenon that lots of people are studying. Oh, this thing is taking a video of me drinking water. Did you guys find the active ratchets paper? Okay, good. Say again, sir. And that's the second paper. The one that I have is the first paper, the theory. Leonardo found the second paper, the one that's cooler, the bacterial ratchet paper. But I wanted to subject you to the theory paper. Oh, the controlling the speed of the agent with light goes in the Science Advances paper. The top line, that belongs in the Science Advances paper. Oh, just copy it, move it to the next category. No, it's correct, but just in the wrong category. Good. By just scrolling through the Angelani paper, you should start seeing at least symbolic similarities between what happened when you were scrolling through the Doring paper. It's actually the same calculation, just rediscovered. Yes, yes, I agree with both statements so far. If I get one more for the Science Advance paper, then I will stop torturing you and go back to my storytelling. Yes, Pablo. In the Angelani paper?
In the PRL.
In the Charlie Doring paper, yes.
I think this is because if the diffusion constant is small and you increase it, you escape from the minima more easily, but if you increase it too much, then it's too undirected.
Exactly. You go so above the height of the barrier that you don't care about the difference in either side. Absolutely. It's perfectly correct. Yes, ratchets controlling the speed of ABPs with light. The nice, cool thing about the Science Advances paper is that you're not building anything. You're just using an external signal to produce whatever you want, which is really cool. It falls under the umbrella of self-assembly. Okay, let's continue on our journey. These are my one-line summaries. The first paper contains RTPs. So they are run-and-tumble particles. They are not ABPs. And it's in a ratchet potential, but calculates the same thing as Doring did. The only difference is the stochastic process. The Markov process used here is run-and-tumble, while Doring uses one-step Markov and things like that. And the Science Advances paper, I really like it that it marries MIPS and rectification to make ratchets on the fly. So it takes two ideas, brings them together to produce a current. So it's really cool. Okay. Now, I'm switching to story 2. Story 2 is putting ABPs inside boxes. I want to put ABPs inside hard boxes. The walls are stiff and cannot deform, and I'm going to put ABPs inside the box. The box has some characteristic size L, and if the characteristic size of the box is such that the time taken for a self-propelled particle to transit the box is short compared to the decorrelation time in the rotational diffusion, then the particles will mostly be at the wall. Yes? Everybody is okay with that? I just run into the wall and then I stay at the wall because I keep pushing on the wall. Right? This is called boundary accumulation of self-propelled particles. And again, there is a treasure trove of literature out there where people have looked into this. And I want to spend a moment talking about what happens at curved walls. I run into a wall, and nu hat in this picture is the direction of self-propulsion. The red arrow is the direction of self-propulsion. And n hat, the black arrow, is the local normal. It's the normal to the curve of the wall, right? So if a particle runs into the wall like that, the wall is going to do exactly the same thing, Newton's third law. It's going to exert an equal and opposite force along the normal, right? So it will kill the component of the self-propulsion velocity that is along the normal, but will leave the tangential velocity. Yes? Okay. Now, supposing that happens, what will happen? This particle will move along the wall, and it will move along the wall until such time as its self-propulsion direction exactly lines up with the local normal. And it will stay there, because the self-propulsion and the wall force exactly cancel each other. So it will find a spot on the curvature where the local normal is exactly with its self-propulsion direction, and it will stay there. Given this fact, so far nothing. This is just Newton's third law at play. I put a bunch of particles inside a curved surface such as this. What I will get is I will get accumulation of these active Brownian particles at the high curvature regions. High curvature regions are the regions where the local normal is changing rapidly, right? If a curve is very curved, that's when the local normal is changing very rapidly. If a curve is flat, there's only one normal. What I'm trying to say is if it was like this is the normal. But if it was like this, the normal is changing from point to point, right? So it can accommodate way more particles than this guy. So the particles will preferentially go to regions of high curvature, and accumulation in regions of high curvature can be calculated almost trivially for the active ideal gas. And it turns out that the density will be proportional to the local radius of curvature. Okay. Now, next exercise. In the same Google Doc, the same Google folder, there's another Google Doc that's called ABPs and walls exercise. This is the same QR code. Find 2 papers that explore the phenomenology of self-propelled particles under confinement, and just put either title and abstract or screenshot or something like that into that Google Doc. Let's look at what collection of papers we are able to come up with. There are easily 100 papers on this topic. So I'm expecting that we should come up with at least 10 distinct papers. This is just to prove to you that this is a very well-studied phenomenon. Put title and abstracts or screenshots. I say put hyperlinks, but now I don't want to click the hyperlinks and open every paper. So I'm lazy. Do you understand what I'm asking you to do? Consider self-propelled particles under confinement. Find papers that discuss this. Yes? Oh, that's the same Google Drive folder. Indication for the papers. So self-propelled particles, confinement, walls, put Google terms like that. Or put wall accumulation. Aggregation. Use words like that. Yes. Oh, I still screwed this up too? Okay. Fix it now. Share. Edit. Apologize for that. Even after I see the name, I can't tell which one is the iguana and which one is the armadillo among the symbols up there. Okay? Yes. The geometric lensing paper is kind of fun. Though I was a little resentful because I felt like we had put all of that in Yawan's paper already. 2D harmonic trap, yes, good. This is not under confinement, is it? Is it confined? I didn't think Silke's paper had walls. I guess she has walls because she had to get jamming? Okay. Of course, she has. It's a Szabo model, that I remember. And I remember the correlations that they measured, but I didn't remember the wall. Yes, E. coli on curvature, yes. They had to maintain pressure to get the jammed dynamical heterogeneities they were looking for. I didn't remember that they put walls to get the pressure. Oh, what is this? Propelled particles under confinement. Never seen that. What is the AIP paper? Oh, I have not read this paper. They have walls? Good. I stopped reading all the walls papers a few years ago because I was like, okay, I'm not sure I will learn anything that I really want to know, so I just stopped reading them. I was hoping for 10 distinct papers. Where are we at? 1, 2, 3, 4, 5, 6, 7. Good. What is long-time anomalous swimmer diffusion in smectic liquid crystals? Does this have walls in it as well? The Hartmut, Lowen, and Wensick paper? It has walls in it? It does? Okay. Because the title, I mean, the abstract doesn't tell me anything about walls. Okay? Good. This is just to give you a flavor. A lot is known about the fact that as self-propelled particles accumulate at walls, the nature of the wall changes the phenomenology of the system, and so on. Let's pause for a moment and think about how crazy this is. Why is this crazy? We pretend that we can do thermodynamics for active matter. We pretend that we can do statistical mechanics for active matter. But if there is so much determined by what the wall is, how can the system ever be extensive? An extensive system, by definition, should be insensitive to what you do at the wall. Right? Surface is negligible compared to volume is the definition of an extensive system. A wall cannot play a role in its physics. But active matter is not extensive. What you do to the wall changes the game. So this is a very important feature of active matter, and we have only just begun to explore the role that this phenomenon plays in various phenomenology that we are interested in active matter. So that was the point of that exercise. Now, let's go to, now I'm switching out of all of this pedagogy. This was all supposed to be pedagogical, though looking at your faces, I'm like, I don't know what happened here. But I'm going to talk about I'm going to tell you a story that is a research story from a recent paper. So I'm launching into my third story, which is, where is this? Yeah. ABPs and bags. This is also active Brownian particles under confinement, but I distinguish bags from boxes by saying that these are deformable. I want to put ABPs inside containers that are deformable, okay? Oh, now that I'm looking at Antonio, and I did not put the Hexbug movie I wanted to show. If we have time, I'll go pull it up from somewhere else. Okay. What I'm going to consider over the next 15 minutes or so is I'm going to consider self-propelled particles. They may be ABPs, or they may be rods, or they may be semi-flexible polymers. You will see simulations from all three categories. They are just ABPs as far as isolated particles are concerned. This is all they are doing. The shape of the particle only enters when you calculate the excluded volume interaction. When two of them collide, you calculate the excluded volume interaction to figure out what the forces are. And if you're a rod, you're going to turn. If you're a sphere, you're just going to stick. And if you're a polymer, you're going to bend. So only excluded volume knows about the shape. Activity doesn't know anything about the shape. That's the circumstance we are going to consider. And I'm going to put them inside deformable deformable boundaries that are basically made of balls and springs. So you make a deformable boundary by linking balls and springs on a computer, and you give next to nearest neighbor interaction so that you can give rigidity to your polymer, and you can control the bending and the stretching and stuff like that by changing the parameters of the bond. And when I go to 3D, this will be a 3D triangulated mesh, again balls and springs, and I can tune the bending and the stretching modulus of my elastic membrane that I formed with these balls and springs by just changing the bond potentials, the dihedral potential, the local potential, and stuff like that. Okay? So that's the game we are going to play. And I want to start in 2D. So this is rods in a rubber band. And the function I want to examine is transport. I'm not so much interested in the statistics of how the rods are arranging themselves inside the membrane and things like that. I don't care about that. What I care about is how does the membrane as a whole move around. Okay, so I want to consider the transport of one of these rubber bands that contains these self-propelled particles. And I want to begin by showing you 3 movies. Let's just get oriented. The movie on the far, whatever this direction is, has V0 equal to 3, that's the activity. A equal to 10, that's the aspect ratio. It's telling you what is the length of the rod. Okay? And the K is the stiffness of the rubber band. So you have a very floppy rubber band, you have a somewhat stiff rubber band, and you have a rigid rubber band over here. K is the bending energy, it's a bending modulus. It's not quite an energy, but it's a modulus. Okay? So I'm changing the stiffness of my rubber band as I move from this side to that side. Everything else is fixed. It's a rod of aspect ratio 10, moving with a speed 3 in Peclet number units. Okay, now let's watch the movies. The movies are just for you to get a sense of the phenomenology. In the very floppy case, it doesn't appear to be moving much, and there appear to be 2 clusters, and sometimes it gets screwy and things like that. The one in the middle is moving a lot, yes? And the one on the side is moving a little bit, but the clusters don't seem to live for very long. They are wandering all over the place. Is that the impression you get by watching these movies? Okay. Now, let's think about how do I go about understanding this phenomenology. I have different organization of active particles in all 3 movies. Would you agree? There are 2 apparently stable clusters in the floppy rubber band. There is only 1 cluster that seems to live forever in the middle rubber band. And there is all this transient clustering happening in my 3rd rubber band. So I have different organization of active particles as I change the stiffness. The vesicle as a whole acts as a persistent random walker. So if I decided to tag the center of mass of my vesicle, the rubber band, and I watched it as a function of time, it obeys a persistent random walk. of some sort. Okay? What I'm going to use as a parameter to try to understand this phenomenon is diffusion. If I took a persistent random walker and decided that it was a self-propelled particle, I would partition the random walk into a V0 and a dr, right? The V0 would be the persistent part, and the dr part would be the one that randomizes the motion. It turns out for these guys, if you try to do this V0 dr partition, it's not robust. Every realization of your simulation gives you a different partitioning between V0 and dr. But if you calculate the diffusion, if you calculate V0 square by dr, that is, you measure the asymptote for the diffusion coefficient for the vesicle, that's well defined. Irrespective of realizations, irrespective of noise, you get a good number. So what we did was we used the diffusion coefficient and said, if the diffusion coefficient is large, the vesicle is moving a lot. If the diffusion coefficient is small, the vesicle is moving less. Okay? We establish the persistence, but we don't use V0 dr language, just because V0 and dr were too noisy to tell a story. Now, what I want to do is I saw these movies, and I want to understand these movies. So I want to try to walk you through what it means to understand these movies. So always, whenever you see a new phenomenon, you first establish Bayesian priors. What do I mean by that? Establish in your mind the things that you already know that may be relevant to what you're seeing now. That's how you approach reading anything. You read a paper or you see somebody's data, you look at it and say, what does this remind me of? And make a list in your head. Right? So let's make a list. I know particles accumulate at walls, right? And I know that particles will accumulate in regions of high curvature because of this story that I told you. And what I have extra now is I have feedback. Once there is a cluster, the curvature will increase where the cluster is because the rod is pushing on a deformable boundary. Right? So there's feedback. When there's a positive feedback which says that, oh, the cluster will form in a region of curvature, then the curvature will increase, that will recruit more rods because the curvature went up. So I have a positive feedback mechanism here because of the deformability of my membrane. And now I want to unpack this positive feedback and try to understand the phenomenology. What I should expect is larger, longer-lived clusters as the boundary becomes softer, based on this argument. Think about it for a moment. The first three statements imply the last statement. Think about it for a moment and yell at me if the logical thread is missing a piece. It's not missing a piece inside my head, but my head has been seeing it too long. So there might be pieces missing that you should point out to me. Okay. Either everybody is buying what I'm selling or you guys don't care. So let's see if my expectation is held out. So I'm thinking the softer the rubber band becomes, the higher the diffusion coefficient will be. That's my expectation. So let's look at the data. I'm looking at the data for a number of different rods of different lengths. If you look at the data L, I start from a sphere and work my way up to a rod of length 5. Okay? And what I find is, okay, for the L equal to 4.5 case, my expectation is borne out. As this curve, as this is stiffness, so this is the stiffest rubber band and this is the softest rubber band. And so if I go this way, the diffusion is going up. Okay, so if I go to rod length 4.5, my expectation is borne out. But for these lower rods, these not quite rods, these spheres and dumbbells and things like that, my expectation is wrong. What did I do wrong? What did I miss in the argument I laid out in the previous slide? Why am I not seeing an increase in the diffusion coefficient, increase in the transport as I soften the rubber band? Yes. They are either spheres or very teeny tiny dumbbells and things like that. No guesses? They align less. The wall is failing to exert enough torque to to align these rods. That's exactly right. Because the stiffness dependence is so weak because active forces cannot be realigned. If you are a sphere and you go run into the wall, there is no way for you to turn around and recruit more guys and line up nicely and increase the diffusion because you can't turn. So you need a longer aspect ratio for my story to be valid. If I was dealing with spheres, it doesn't matter at all. You cannot get an enhancement of diffusion by making the rubber band floppy. The sphere does not care about the floppiness of the rubber band. So when you do it for spheres, this is for same initial condition, different realizations, different noise realizations, different details. But the trajectory of the vesicle looks exactly the same because it doesn't care. It doesn't care whether your rubber band is soft or hard or what. Now, this is for these marginal rods. What I mean by that is it's kind of shameful to call them rods. They have aspect ratio 5. For a thing to behave like a rod, the threshold aspect ratio is 7. This is just a rule of thumb you can use. In 2D, for you to get sufficient alignment and sufficient nematic or polar ordering in your system, the aspect ratio is 7. That's a good rule of thumb. Anything less than 7, it's marginally a rod. It doesn't behave like a rod. It behaves like some fat guy. So the marginal rods do show some increase in diffusion, exactly like I expect. And now I move on to really long rods. And I find this. Again, my expectation is wrong. I said as the rubber band becomes floppier, the diffusion should go up. But instead, what I'm finding here is a Goldilocks value of stiffness. There is a particular value of stiffness at which the diffusion is very large, but it's smaller on either side. And this large enhancement, look at the scale bar. This is 2.5 and this is 80. It's an enormous enhancement. We're talking about orders of magnitude. Okay, so there's a very sweet spot where stiffness and rod activity cooperate in such a way that the vesicle moves like a bullet, moves really fast. Okay? So I want to understand this sweet spot. How did it pick out a particular bending stiffness as its favorite value to move around? How did it do that? Let's go back and look at the movies again. The middle one is the Goldilocks movie. The middle one is the one that has this 100-fold enhancement in the diffusion. The two sides are the ones that have less diffusion. Now we have aspect ratio 10 here, so we are above the threshold for alignment behavior. Would you agree that the middle movie is special because there is one cluster that seems to be ridiculously long-lived? It's not yet gotten to the one cluster yet, but in a minute it will form a one cluster, and then that cluster will live forever. At least as long as this movie is. Yes? Okay. So the game is, I get a single long-lived cluster inside my rubber band for some particular values of bending modulus. Okay? So now the game is to find what are the circumstances under which I will get one long-lived cluster in my rubber band. So let's try to see how that works. I'm going to give you a cartoon picture. I'll put the references for the paper where the calculations are, but I'll give you a little cartoon picture of how to think about it. So imagine a self-propelled rod. It runs into the wall. You all know at this point that if I run into the wall, I will turn parallel to the wall. Yes? That's what will happen with the force and torque exerted by the wall. It will become parallel to the wall. So when one rod runs into the wall, it will become parallel to the wall. Now let's imagine that two rods are parallel to the wall, but they meet at the wall like this. This recollision will result in the rods becoming like this, normal to the wall. So step one, I run into the wall and become parallel to the wall. But other guys are also going to running into the wall and becoming parallel to the wall, so I'll meet somebody at the wall. And when I meet somebody at the wall, I will become perpendicular to the wall, because there is no other way for both of us to be there. And basically, you start accumulating guys that will start becoming like this. Does that make sense? Two guys meet. They meet here, and they form this. Then another guy comes. He also joins. The other guy comes. He also joins. The cluster starts growing. Right? Now, this collection of rods is happiest at a flat wall, where all of them are pushing into the wall. Wall is pushing back on them equally. Right? If you put them at a curved wall, they are not going to be that happy, because the curvature is pushing them closer than they actually want to be. So that results in a shear stress on a large cluster. If the cluster becomes too large, it's unhappy. The shear stress breaks the cluster. So you combine these two to figure out the optimal size of the cluster by asking, these are the two mechanisms. The first mechanism is that if the rods meet, they'll become perpendicular and form a cluster. The second mechanism is as the cluster grows, it becomes less and less happy because it's getting sheared by the curvature. So you balance these two effects and you find the ideal size of your cluster, and that ideal size will tell you, you use the ideal size to figure out what is the relationship between stiffness and activity and aspect ratio and number of particles inside your bag. That will give you this ideal cluster. And it turns out to be this relationship that I have here. I'm not writing out the math that leads to this relationship. It's in this paper that I cite here from last year. And oh, I thought I had one more on this. Okay, apparently I'm moving to story 3B. That was all about 2D, things in a rubber band, right? Now I want to put them in a vesicle. So these are things in 3D, same rods, self-propelled, everything is the same, only now we are in 3D. And I hope you will be able to notice that depending on the wall, here I'm keeping the stiffness fixed. In the particular phase diagram I'm showing you now, yes sir?
So just a question. This is all dry active matter.
Yes, yes, absolutely.
So I wonder if you do an experiment, it's never going to be dry. A vesicle is going to be filled with liquid. So would wet active matter just change a prefactor in your?
No. So there are some experiments where people have put E. coli inside GUVs, right? And we have put microtubules and kinesin motors inside GUVs, things like that. Okay. And what happens in those circumstances is that there's serious backflow. And that serious backflow prevents these guys from polar alignment. They actually align pneumatically. And when they align pneumatically, the physics changes. So bacteria or suspensions, anything, even if they are self-propelled, they don't behave like self-propelled. They behave like pneumatics. They behave like Julia's system. So the phenomenology is actually very different. This is only good for hex bugs.
Yeah, and the ones you mentioned are pushers. I guess pullers would behave differently? Or it would be the same? Okay,
so the pullers and the pushers at the wall are complicated. I don't know the answer to the question. What I'm saying is definitely true for pushers. For pullers, the same phenomenology holds, namely that the backflow is going to change things into pneumatic, but the stability of a puller pneumatic is different. So I don't actually know what the cluster size organization is. I have not done it, nobody has done it that I know of, and I haven't seen an experiment, and off the top of my head, I can't guess what will happen. So I'll get one of my friends to put clammy inside some GUV and see what happens. Yes, sir?
Yeah, I have a, just trying to make sure I understand the argument of the three examples that you had before.
Yes.
And the middle one had, if I understood correctly, it had an optimal curvature compared with the cluster size so that it would have just one big cluster.
Exactly.
If I put more particles there,
no, then you'll break it.
It breaks. Okay,
yes, exactly. Yeah. That's exactly what happens. So you have to, once you pick the rubber band stiffness, you have to optimize for the activity and the number of rods you put inside it. Or if you pick the number of rods and activity of the guys inside, then you have to pick the rubber band stiffness appropriately to make one cluster.
Got it. Yeah.
I thought I had one more graph, which was a scaling collapse that tells you how these different parameters relax to each other. Sorry. Yeah. So now you can do this in 3D. And in this case, I'm not changing the stiffness of the rubber band because, well, there is another plot in the paper that just changed the stiffness of the rubber band. But here I want to focus on what happens as I change the length of the filament self-propelled filament I put inside my vesicle, and what happens when I change the number of rods I put inside the vesicle. Those are the two axes. Do you see? This is the filament volume fraction. So I'm adding more and more self-propelled particles as I go this way. And on the other axis, I have aspect ratio. These are becoming longer and longer as I go this way. And you'll be able to see that the very long guys, even in the dilute regime, form this nice self-organized equatorial ring. This is a feature of self-propelled particles on spheres. And when that equatorial ring becomes strong enough, by adding more rods, you get this oblate object, this squished red blood cell-like object that is formed. But if you reduce the aspect ratio and made your rods shorter, you instead get these capped states where you get these dumbbell-like structures, or you get these polygonal structures and things like that. So there are interesting shape transformations that you can observe depending on the number of particles you put in there and the length of the particle. Is it a very long particle? Is it a very short particle? Things like that. We've also changed the stiffness of these guys, the stiffness of the long particles we put in there. This is all at fixed rigidity. And this whole zoo of things is there in this Nature Communications paper from a few years ago. It's a big phenomenological zoo. And then we've analyzed a few different phenomenologies and tried to explain exactly along the lines of how I did the 2D explanation as to how do people meet at the surface, and how they organize, and how that depends on the stiffness and the activity and things like that. So if you're interested, I suggest that you look at that story. Okay. Yes, sir.
Do you find in some run and rotate states the vesicle?
Oh, they probably all
moves and rotate. And
yeah, I'm sure. So there are. What we did in this study is we just picked the function of shape change, and we kept bringing the simulation back to center of mass so that we didn't care where the vesicle was moving. But if you do care about where the vesicle is moving, you do get these bullet vesicles that keep going in some direction and things like that. Yeah. But I didn't put that result in this paper because it became too hard to write. I couldn't tell the story. Yes, sir.
It's just about the terminology, because for me a vesicle is a 3D closed object. But if you want the vesicle to move, then bacteria need to walk on something.
Oh, yes.
So in an experimental vesicle, it wouldn't move.
An experimental vesicle can also move if there is a differential slip between the outside of the seed ice vesicle and the inside of the seed ice vesicle. That results in some treadmilling, rolling, whatever kind of motion. So there is some possibility, but this is dry. Yep. So here I have a magical momentum sink that makes all of these things go away. So the vesicle, have you seen the E. coli in GUV paper? There's a paper by Takatori and Sahu, which does a good job of explaining what happens in the wet case. So, and there's also another paper by, well, I can't remember right now. Takatori and Sahu is a good starting point. They probably cite the other paper as well. Okay, so what have I done so far? I told you some random stories about ABPs. The first part of the story was still ideal gas, right? None of them were talking to each other. They were only talking to the wall, right? That's what an ideal gas is. An ideal gas is a system where the particles don't see each other, but when they run into the wall, they bounce. Even your PV is equal to NKBT ideal gas, that's what it does. The particles don't see each other, but when they run into a wall, they bounce, right? And we looked at what do walls do when these particles are not interacting, right? Then I turned on interactions and made my wall flexible, right? I turned on interactions so that there's excluded volume interactions between my particles, and I made the walls floppy so that they could bend. This could give me feedback between what the active particles wanted to do and what the vesicle wants to do, and this feedback leads to some organization and some structure, and these things can be harnessed for function. You can make things do stuff you want it to do, right? Active matter self-organization is only interesting if you can harness it for function. Otherwise, only StatMech people will care, right? And these are two functions that I have illustrated today. One was transport. How do I get things to move fast or move less? And the second was shape transformation. How do I take a spherical vesicle and use self-propelled particles to shape it into an ellipsoid or shape it into a oblate guy or something like that, right? Okay. And in the previous paper, in the 2D paper, I wrote in the discussion a long set of ideas associated with what else you can do with this model. And I encourage you to look at that discussion because you can use this for all kinds of, I am going to build a collective motility object out of single motile objects that don't actually talk to each other except through physics, right? There is all kinds of things that can be done. I cite a bunch of collective robotics literature and things like that in the hope that somebody else will do it. I don't want to do this, but I want to know the answer. So please read the discussion section of the previous 2D paper, and when you solve the problem, let me know so that I'll just hear the answer. Okay. Now, I want to change gears. I actually did not want to talk about MIPS, but we discussed MIPS in various conversations during the lecture. So I want to make a few remarks about MIPS just to place context to how my head is working. This work is now about 12 years old, right? So my understanding has evolved over the last 12 years by reading all of the papers that everybody else has written. So I just want to reflect on it for a moment. Just for completeness, in case anybody has not investigated these systems, I want to recall what I mean by MIPS. So I imagine putting a bunch of spherical ABPs, circles, inside a periodic domain, excluded volume interactions, no torques. If two guys run into each other, they keep running into each other until one of them turns, right? And I vary the packing fraction, which is on this axis, and I vary the Peclet number, which is on this axis. So this is activity, and this is density. And what I find is this. So this is activity, and this is density. When I have zero activity, as I raise up density, all I get is a hard sphere liquid, hard sphere gas, hard sphere crystal, though I don't get the crystal quite here. And on the activity increasing axis, at low density, nothing much happens. It looks like a gas. But at high densities and high activities, you get this phase separation phenomena, which is what we have been calling MIPS. Now, the theoretical paradigm that most people use to understand MIPS is this phase separation paradigm, right? So what you say is at low Peclet number, if I histogram the density in my box, I will get one peak. But as I increase the Peclet number, the fluctuations increase, the peak broadens, and eventually the peak splits into a high density peak and a low density peak. That is your standard metric for phase separation. And you ask when does the split happen, and you draw the binodal line. That tells you where the MIPS process is happening. Okay? Now, this is the phase separation consideration. Let's switch for a moment and ask what are the kinetics considerations that allow this phenomenon to happen. Let's imagine that I have a cluster of self-propelled particles. Then the kinetics of this cluster are basically there is some rate at which particles join the cluster, and there is some rate at which particles leave the cluster, right? And if I was doing equilibrium, the rate at which particles join the cluster and particles leave the cluster is given by some free energy difference between the high density and the low density phase. Yes? Detail balance. The rate will be determined by the free energy difference. But in the active case, you have a breaking of detail balance. So the on rate is set by the self-propulsion velocity and the density, but the off rate is set by the rotational diffusion coefficient. It's about how quickly you rotate away from the direction in which you are pointing, right? From these kinetic considerations, you can realize, exactly like in the previous two cases, it's a question of comparing the rotational diffusion time to some other time in the problem. Okay? In the first case, in the rectification case, we considered the rotational diffusion time and we compared it to the time taken to transit the barrier. And when the time taken to transit the barrier is short compared to the rotational diffusion time, you got rectification. In the case of wall accumulation, we compared the time taken to transit the box to the rotational diffusion time. And when the time taken to transit the box is short compared to the rotational diffusion time, you get boundary accumulation. It's exactly the same thing that's happening here, but the time scale that you want to compare to the rotational diffusion time scale is the collision frequency. So 1 over the collision frequency is the time scale. And you ask, when does the 1 over collision frequency time scale become short compared to the rotational diffusion time, and that's where the clustering will happen. Okay? That basically gives you an estimate of where this MIPS, where should I go from a gas to liquid, right? And that gives you, says that the collision frequency should be equal to dr. Now, what is the problem with figuring out the MIPS criticality and all of this stuff in numerical simulations of actual interacting particles? So what I wrote on this slide is collision frequency, self-propulsion speed, size of the particle should be proportional to dr. Okay? Now, to make this equal, I need a number here, right? And this number, away from the critical point, turns out to be rather easily determined from numerics. But as you get closer and closer to the critical point, what number you find turns out to depend on all kinds of details. So what I identify as the critical point may be slightly off from where the critical point actually is. And since I have some uncertainty in where I'm placing the critical point, where I place the critical point and how I compute the critical fluctuations, the scaling exponents and things like that, turns out to depend on the details. And therefore, that's why we have not managed to agree. Because after I saw Thomas's experiments and Thomas's results, I went back and looked at my data. And I realized that, oh, this is all super sensitive to the details of what one is doing inside the simulation. The universality of this proportionality constant should be universal at the critical point. Because the critical point should not care about whether you're doing WCA with 1 coefficient or you're doing WCA with 2 coefficients and nonsense like that. But it turns out to care. So we need to go to larger system sizes or something else. I don't know what is missing, but it's hard to reconcile the data from different pieces. Yes. Oh, sorry.
So this parameter is proportional to the amount of avalanches that happen,
yes.
right?
Yes.
And near the critical point, you expect to have a lot of fluctuations.
Yes.
So that's why this parameter changes a lot?
Yes. So it's very, very sensitive. So you need a lot of statistics to pin down that parameter. And you never get enough statistics as far as I'm able to see. So it depends on the details of everything that you have.
Okay, but in principle, the average of this avalanche is universal?
Presumably.
Okay.
Yeah, we just haven't established it because all of us who are sitting on the data are not able to agree on it. But I expect it to be. Yes. Okay. So those are just some comments on the MIPS. We do not have time for philosophizing. I just put, do we have time for philosophizing to decide whether we will have time to talk about it or not? So we won't. I'll stop here. And just because I promised Antonio, let me see if I can find my Hexbugs movie that I wanted to show. It's not my experiment, but I like the experiment and I want Antonio to try it. Where would I find it? I'm going to stop share for a moment so that you guys don't see all my top secret slides. I apologize. I know I'm done talking. You guys can leave if you want. I just want to show the Hexbugs movie. Oh, the Hexbugs movie is not here. So these are the Hexbugs movies that I wanted to show. I assume everybody has seen this paper? Oh, I am watching it and enjoying it and not showing you guys. Sorry. I pulled up an old talk that I have this movie in. This is a rubber band. It has rods inside it. And it organizes into 2 clusters. And it goes through a barrier. One more. Because these people made a big high art of doing these things. I'm assuming everybody has seen these movies. It's going to open the door. And one more. This is, they call it the hex bugs Roomba. It's eventually going to push all of these spheres into corners. It's boring. It's taking too long. It will eventually get there. Wait. See. Do you see? Once you put them inside rubber bands, it just goes and pushes everything into one corner. And I can tell you how to make this navigable. If somebody makes a rubber band with things inside it, I'll tell you how to make it navigable by sticking a magnet on just one guy, because it's a very easy theoretical calculation to do. So there's something fun for people to try. This is, I don't know. Oh, this is also the Roomba movie. Okay. I just wanted to show that for fun, but I forgot to put it in this morning. Yes, you are all liberated. We are done.
Any more questions for Aparna?
Okay.
If not.
Right. Tomorrow will be again a lecture like today, but no pedagogical exercises. I will be just talking about things that excite me. It's an overview of active matter from Vichek up to whatever games we are playing right now. Okay? So you can take a nap in tomorrow's lecture. My headache is making me very stupid, so this lecture was a little rough.